So thank you very much, Thomas, first for your invitation to come here. It has been beautiful to visit Kiel and to see such outstanding scientific work here. It's been a great day. I've really enjoyed it. And thank you, ladies and gentlemen, for coming here. Thomas has given a very kind introduction, so I'm gonna get started. So I'm going to discuss early life and its role in human development. And maybe the subtitle is Our Missing Microbes, because I hope to show you that these events are related. So what I want to discuss, and as Thomas told you, I'm a medical doctor and a microbiologist as well. I want to discuss why we have seen a shift in diseases essentially since World War II. Here on this slide are 3 diseases that have gone up in epidemic proportion since World War II, and the list goes on. Maybe there are 10 major diseases that have gone up. So why have these diseases all risen? Is it just chance? 10 diseases, are there 10 different causes? Or perhaps there's an underlying cause for all of them, and I'm going to discuss that question. I'm going to focus in the beginning of my talk on obesity. And this is a slide from data from the Centers for Disease Control in the United States, looking at trends of obesity in adults in the United States, which we could consider a form of changing physiology. The important points are that in 1989, there is no state in the United States where more than 14% of adults are obese. But by 2010, there is no state with less than 20% obesity, and the national average is about 30%. So what's remarkable to me is that it is happening everywhere in the United States, and the difference between the first map and the last map is only 21 years. So I think you're all familiar with the epidemic of obesity in the United States. But it's not just in the United States. Here are data from the Netherlands over about a 30-year period looking at the proportion of adults who are overweight. You can see the same kind of trend. Here's percent of adults who are obese in 10 countries. Here's the US. Here's Germany, not too far behind. Some of the other countries that have very high rates are Mexico, Saudi Arabia, South Africa. These are developing countries, or countries that we might not ordinarily associate with obesity. Here's obesity in Germany by age group. And you can see the trends, how obesity becomes more common as people get older. These are 2008 data. Now, it's clear from many studies that adult obesity begins in childhood. So what are the trends on childhood obesity? Well, these are also data from the NHANES national studies over about a 35-year period in the United States, looking at obesity trends in children by age. And you can see the trend is in the same direction, and it's beginning in the youngest group, age from 2 to 5. Here's childhood obesity in Germany. I recently was studying the internet for slides about Germany. But basically you're seeing, here we're looking at a 36-year period. You're seeing the same kind of trend. This is telling us where the epidemic is going. Now, recently there were data presented looking at obesity trends across the world. Here we're looking at children under the age of 5, percent overweight and obese. And in developed countries, in 2015, it was about 12.5%. But in developing countries, and we don't think of obesity as a problem in developing countries, it's about 7%. So it's lower, but it's not so much lower. And in fact, if we consider where the developed countries were and where the developing countries are, it suggests the idea that they're lagging about 30 years behind the developed countries. Now, those are based on percentages and trends. But here are data on numbers of children under 5 who are overweight. And in 2015, it was estimated that there were about 50 million children who were overweight in the world, under the age of 5. Most of them are in developing countries. Obesity is a giant epidemic in developing countries, and the trends indicate it's going to get worse. Obesity isn't the only disease that is rising. I mentioned others. I also want to mention inflammatory bowel disease, because there's evidence that it's been going on for some time in the West, and we're seeing this later curve happening in some of the newly industrialized countries. The same kind of time lag. In this case, it's a longer time lag, but still same trend, but lagging. Now, in order to understand this, I want to introduce 3 important concepts. The first concept is evolution. And this slide is from the work of our mutual friend Howard Achman and his colleagues, looking at evolutionary relationships of wild hominids. Here on the left side is a phylogeny of wild hominids. And there's a branching pattern, a topology, that's based on the hosts. And if we go to the right side, we look at the phylogeny based on the fecal microbiota. And we can see that there is a lot of congruence between these 2 phylogenies. So this congruence is strong evidence for the vertical transmission of the microbiota, and it is also evidence that is consistent with the idea of coevolution of microbes and hosts. Here we're looking at a scale of about 8 million years. And there are other studies in mammals suggesting it's going back 50 million years or 100 million years. Now, I want to contrast this slide of our 8 million years of shared evolution with this slide, where we're looking at the prevalence of Helicobacter pylori in the United States. And Helicobacter pylori, until recently, was the dominant organism of the human stomach, in which virtually everyone was positive. And this slide, also from NHANES data, looking at males and females, here it's organized by year of birth. And we can see that people born early in the 20th century frequently had Helicobacter. And when they're born later in the 20th century, they infrequently have Helicobacter. These are cross-sectional data, but longitudinal data show the same kinds of trends. It is clear that there's a birth cohort phenomenon, that Helicobacter pylori, which has been present in humans for at least 100,000 years, over the course of a century, is rapidly disappearing. Now, the second concept I want to talk about is age window. And maybe if I go back to this point, I can just say that we have a conserved coevolution that is rapidly changing. So next point is age window. And this is work by Tanya Yatsenenko and colleagues working in Jeff Gordon's lab. Maria Gloria Dominguez, Rob Knight, and others contributed to this work. This study and in this panel addresses the question, when does the adult gut microbiome become established? The investigators looked at healthy people in the US, Amerindians in South America, Malawians in Africa. Each circle is the composite of the gut microbiome in one individual. The x-axis is age during childhood. The y-axis is the unifrac distance between children and adults. This is a measure of dissimilarity. And what the data clearly show is that when babies are born, their microbiome is very un-adult-like. And then gradually and progressively, it becomes more and more adult-like until it is. And what's important in this slide is that the transition, the point of transition, is about the age of 3 years. The first 3 years of life is when the microbiome is most dynamic. It's when it's developing. And of course, that's the period of time that babies are developing. They're developing their immunity, their metabolism, and their cognition. Third concept I want to talk about is equilibrium. And this is shown in this slide. This is from work I did with Denise Kirshner, a mathematician. Our idea of coevolution is that colonizing microbes and their host are talking together. There's a series of cross-signaling that forms a kind of equilibrium. Denise showed that this is a dynamic equilibrium. She's shown that it's robust and resilient. What I want to talk about is what happens when it's perturbed, what happens when it's lost. And actually, talking today with Thomas, we can think of the same kind of equilibrium in Hydra and its co-evolved microbes as well. So what happens when it gets lost? And why might it get lost? So I want to come back to this issue of vertical transmission of the microbiota. Maria Gloria Dominguez is going to talk about it at length, but just to remind you that humans, like other mammals, have many redundant pathways for the transmission of microbes from one generation to the other, but we are interfering with it with such things as cesarean section, early bathing, bottle feeding, and of course, early life antibiotics. And it was on the basis of these kinds of issues that over the years we've developed an idea called the theory of the disappearing microbiota, which has two major tenets: that changed human ecology has altered transmission and maintenance of ancestral microbes, which has led to extinctions, and that especially important are microbes usually acquired early in life, since they affect a developmentally critical stage. About 8 years ago, Stan Falco and I enlarged the hypothesis, which is shown here, the effect of maternal status on the resident microbiota of the next generation. Our view of ancient moms is that they had an ancient microbiota, which they would then give to their offspring vertically. If they lost microbiota before the next generation and couldn't get it back horizontally, then the next generation would be born at a deficit, and so on and so forth. So in this view, the loss of microbiota is cumulative across generations. The conduct of one generation affects the next generation. There's already evidence that supports this view. Here we're looking at the prevalence of Helicobacter in 3 generations in Japanese families, and you can see the same kind of step that we have postulated. Maria Gloria Dominguez is going to, in a few minutes, will show her work comparing total intestinal diversity between a hunter-gatherer group in the Amazon, tribal people, and people in the US, showing the same kind of step. So we have support for this idea of the cumulative loss of the microbiota. So what could be causing it? I think there are many factors, but I'm going to, for the rest of this talk, I'm going to focus on a single factor, and that's antibiotics. All of us have grown up in the antibiotic era. We know of antibiotics as one of the greatest discoveries of mankind in the 20th century. Antibiotics have saved countless numbers of lives. It has revolutionized all of medicine. Here's Fleming recreating the discovery of penicillin. But because antibiotics are so beneficial, doctors and others have been using antibiotics more and more and more. So how much more? Well, recent data from the World Health Organization counts about 73 billion antibiotic doses annually across the world. That's 10 doses of antibiotics for every man, woman, and child on Earth every year. In the US, the CDC has counted 258 million courses of antibiotics. That's 833 courses per 1,000 people. That's 5 courses for every 6 people in the United States. Extrapolating from the CDC data, in the US, children, by the time they reach 2 years old, have had nearly 3 courses of antibiotics. By the time they're 10, they've had 10 courses of antibiotics. In pregnancy, just before the intergenerational transfer of microbes, more than 50% of pregnant women in the United States are treated with antibiotics. And there are many exposures from the use of antibiotics on the farm, but we don't even understand the scale of that. Those exposures are low dose, but they are very frequent. Here, just as an example, here's the fraction of Danish children receiving antibiotics in the first 2 years of life. I couldn't find data from your state, but Denmark is close. pretty stable over 13 years. More than 70% of Danish children are getting antibiotics in the first 2 years of life. And I want to contrast that with a study from Denmark looking at the likelihood of inflammatory bowel disease in Danish children by early life antibiotic exposures. Here's the reference group, no antibiotics. The more courses of antibiotics, the higher the risk for developing inflammatory bowel disease in the first 10 years of life. That's not even the peak of IBD. The data from Denmark suggests that every course of antibiotics contributes an increase of risk of IBD of 14%. So our big picture hypothesis is that antibiotics are having an impact on long-term physiology through microbiota changes. That in health, the ancient gut microbiota is talking to developing cell populations, and the nature of that conversation is affecting early life cellular development in adipose tissue, muscle, bone, immune tissues. This leads to long-term physiology in health. In contrast, our hypothesis is that antibiotic perturbation or other kinds of perturbation are changing the gut microbiota. They're changing the conversations with developing cells, and these are changing tissue development, leading to an altered long-term physiology. So this is the hypothesis. Now I want to go back to this issue of obesity, because now for 70 years, farmers around the world have been performing a very big experiment. They have found that if they treat their farm animals with low doses of antibiotics, they will gain weight. This is what is called growth promotion. And the reason that farmers do it is because it works. They have found that this subtherapeutic antibiotic treatment, which we abbreviate as STAT, increases the growth from chickens to cows, big swath of vertebrate evolution. It seems to be true also for insects. They have found that virtually every antibacterial agent that has been studied has an effect, regardless of its chemical structure, class, target, or spectrum. Antivirals do not have an effect. Antifungals do not have an effect. It suggests that it is directly because of their antibacterial effect. Importantly, the farmers have shown that the earlier in life the antibiotics are started, the bigger the effect on the growth rate, the bigger the effect on feed efficiency, the conversion of food calories into body mass. So it's difficult to study farm animals, but we can study mice. So we began a series of studies in mice in which we would give mice antibiotics or not. We would examine the properties, look at the microbiome, and seek relationships. So now, for the rest of the talk, I'm going to show you data from a number of different experiments. So the first experiments in this area were conducted by Il-Sung Cho when he was a postdoctoral fellow in my lab. Il-Sung gave 4 different regimens of antibiotics to mice using the midpoint of the FDA level on the farm, and compared here percent body fat in the control mice and in the mice that received antibiotics. And you can see that there was an antibiotic effect. This was our first evidence that antibiotics could change metabolism in the mice, that could change body composition. Il-Sung showed many other things, but I want to go on to some other experiments. So in the next experiment, Laurie Cox, who was a graduate student in the lab, did an experiment that we called FAT-STAT. She asked what happens if we combine a high-fat diet and the STAT, that's the subtherapeutic antibiotics, on body composition. She studied control mice, or mice that received subtherapeutic doses of penicillin. All the mice got normal chow, and at week 17 of life, half the mice were put on a high-fat diet, and half remained on the normal chow. She studied both male and female mice. Here we're looking at what happened to total mass, fat mass, and lean mass. If we start with the male mice, looking at total mass, here's the control group. No antibiotics, continued on normal chow. Normal chow with antibiotics, they're bigger, just like on the farm. High-fat diet, bigger still. High-fat plus antibiotics, big-est. Lean mass increased with antibiotics, just like on the farm. Fat mass, big increase on the high-fat diet, even more high-fat plus antibiotics. Female mice, many of the same trends. If we look at fat mass, the average female mouse on the high-fat diet gained 5 grams of body fat. high fat plus antibiotics 10 grams. They doubled the amount of body fat. The antibiotic potentiated the effect of the high fat diet. Up to this point, the mice were getting antibiotics for their whole life. So Laurie did an experiment to ask, are the changes durable if there is only a limited antibiotic exposure? So in addition to no antibiotics or lifelong antibiotics, she gave antibiotics for only 8 weeks or only 4 weeks. Here are the results for morphometry, total mass, lean mass, and fat mass. The black line is the control group. What Laurie found is that all 3 antibiotic regimens increased total mass, lean mass, and fat mass. So 4 weeks of antibiotics early in life was sufficient for the full effect. In another experiment, she gave the antibiotics later. There was an effect, but it was not as strong. Again, confirming what was seen on the farm. The first days of life are the most important. Now, we were interested to what might be the effect on the host. So here, work that Jackie Leung did in Peng Lok's lab, we were looking at the effects of the antibiotic treatments on intestinal Th17 populations. These are cells that are important in immune response and homeostasis in the gut. She looked at the small intestine and the large intestine, and however she looked at it, these levels were down in the mice that received antibiotics. We've looked at this many different ways, and our results are quite consistent, as you will see. So now the question is, what were the antibiotics doing to the microbiome? So here we're going to look at the community structure of the microbiome in mice that were 3 weeks old. And at 3 weeks, there are basically 2 groups of mice. Control mice, no antibiotics, or all 3 antibiotic groups were still receiving the antibiotics at this time. So now on this principal coordinates analysis, the black dots are the controls, the orange dots are the antibiotic-treated mice. They are similar in their distributions, but they're not identical. That's not surprising. One group is receiving antibiotics, the other group is not. Now we're going to go to 8 weeks. And this possibly is the most important slide of all the slides I'm going to show today. At 8 weeks, we have 3 groups of mice. Control mice, no antibiotics. 2 groups of mice that continued their antibiotics, but 1 group of mice that received antibiotics for only 4 weeks and then stopped. And we're looking at 8 weeks. So here's the principal coordinates analysis. Black is the control group. Orange is the continued antibiotics. They are more different than before, not surprising. 1 group is continuing to get antibiotics. But the group that got antibiotics and stopped, their microbiota have normalized. So the effect on the microbiota was transient, but the effect on the phenotype was permanent. So this slide provides evidence that altering the gut microbiome during a critical early stage in development can have long-term consequences. We continued this work by doing a transfer of microbiota from conventional mice to germ-free mice. In this experiment, mice had been given antibiotics or not. We harvested their cecal microbiota and gave these microbiota to germ-free recipients who we then observed for the next 5 weeks. They did not receive any antibiotics. So here we're looking at total mass, lean mass, and fat mass. The black line is the group that received the unperturbed control microbiota. These mice, the mice that received the antibiotic perturbed microbiota, had an increase in total mass, no change in lean mass, and an increase in fat mass. So this showed that the signal was in the microbiota. The altered microbiota was sufficient to confer the adiposity signal. Next, we asked what would happen to the genes involved in immunity. So in this experiment, we're looking at the donor mice, the mice that contributed the microbiota. We're looking at Th17, transcription factor, 2 cytokines, antimicrobial peptides downstream of Th17. As we've come to expect, these levels are down in the mice that receive the antibiotics. Now the question is, what happens to the recipient mice? And here we see the same kinds of trends. So we have evidence from this experiment that the altered microbiota are sufficient for the immunologic phenotype as well. So this is all, we're looking at markers of disease. And so more recently, Angel Schulfer, a graduate student in the lab, asked, can an antibiotic-altered microbiota affect IBD outcome? Again, remembering the epidemiologic studies in Denmark. So she worked in a model system where she would conventionalize germ-free C57 black 6 mice that were either wild type or IL-10 deficient. These are mice that get the spontaneous development of colitis. And Angel's study design is shown here. She used either the control microbiota or an antibiotic-perturbed microbiota. She gave it to pregnant germ-free mice, and now these mice are conventionalized. And shortly thereafter, these mice give birth to their babies, and now their babies grow up almost for a half a year. So we had 4 groups of mice, 2 genotypes and 2 different founding microbiota. So first, let's ask what happens to the microbiota. So here is the control inoculum. Here's the stat inoculum on this principal coordinates analysis. Now we're looking at the recipients of the control inoculum, the dams and the pups. They're in this quadrant. Here are the recipients of the stat inoculum. These are the wild-type mice. They're in this quadrant. Now we're looking at the IL-10 deficient mice getting the control inoculum. They're in this quadrant. And the IL-10 deficient mice that got the stat inoculum, they're in this quadrant, although you can see there's much more heterogeneity. So this is just dams and pups at day 21 of life. If we just look at the data all the way in the pups out to week 21, you can see that the groups retain their general integrity. We've looked further at the transmission of taxa across generation. This is work we did with Dan Knights and Serena Robinson, looking at the wild-type mice and the IL-10 deficient mice, those that receive the control inoculum or the stat inoculum, comparing the inoculum, the dams, and the pups. One of the first observations we can see is that in these next generations, the microbiota is more sparse than in those that receive the antibiotic-perturbed microbiome than the control. We've looked at the characteristics of this in several different ways. I'll show you one way. Here we're looking at the intra-group variability in dams and pups over time. These are the dams, these are the pups. We're looking across time. These are the same times here. And we're comparing the control and the stat in wild-type mice and in IL-10 mice. In 11 out of 12 comparisons, we're seeing much more variation in the mice that received the antibiotic-perturbed inoculum compared to the control inoculum. And that's similar to what we discussed early today in studies of Hydra. We can also look at the stability of the microbiota using Jacquard analysis. In this analysis, the higher the Jacquard score, the more different, the less stable the microbiota are. And we see in each case, there's less stability with the antibiotic-perturbed microbiota than the control. Here in the dams, regardless of genotype, here in the pups, regardless of genotype. So it's clear that we're sending a signal from the inoculum through the dams into the pups. So now the really important question is, what happens to their IBD phenotype? And that is shown in this slide, where we're looking at colonic histology in the wild-type pups and the IL-10 pups by the inoculum that was received. This column are all those that received the control inoculum, the descendants of those who received the control inoculum. These are the descendants of those that received the stat inoculum. The wild-type mice, they don't have much inflammation. We didn't expect any because they're normal mice. But the IL-10 deficient mice are developing more and more inflammation as we expect, but it's much greater in those that received the antibiotic-perturbed microbiota. And we can look at it in several different ways, and the results are consistent. So these data show that the inheritance of the antibiotic-perturbed microbiota are having an effect not just on the dams, but on the pups. Pups never saw antibiotics. In fact, the dams never saw antibiotics. They just saw an antibiotic-perturbed microbiota. So up to this point, everything I've shown you is with our first model, which we call STAT, for subtherapeutic antibiotics. This is the farm model. But human children are getting pulses of antibiotics. They're getting pulses of antibiotics to treat their ear infections and their throat infections. So Yael Noble developed a second model, which we called PAT. Her hypothesis was that a series of short therapeutic dose courses of antibiotics administered early in life will sufficiently change the gut microbiome to alter host phenotypes. From this work, I just want to show you a couple of observations. We're going to look at what happened to the cluster types of the microbiota over time in the mice that were control mice that received amoxicillin, the most widely used antibiotic in childhood, or tylosin, a macrolide, the second most widely used class of antibiotics, or a mixture. We're looking across the time point of the experiment, and informatic work by Alex Alexienko suggested that there were 4 major cluster types in this population of almost 350 specimens. And I can summarize it by saying that in the control mice, in early life, they have a maternal microbiota. It looks like their mother's, until they're put on a high-fat diet, when there's a big change to this high-fat diet community structure. Amoxicillin looks like control, but there's some perturbation. But the macrolide shows a lot of perturbation, and it only finally does the high-fat diet with its strong selection normalize it. So this indicates that the effects can be long-standing. The antibiotic ended here, and this is months later, and also that all antibiotics aren't equal, that the macrolide is having a much stronger effect than the beta-lactam. Now, this work was continued by Victoria Ruiz, a postdoc in the lab, and Victoria continued Yael's work where she studied 3 pulses of antibiotics, but then she asked what happens if she just gave a single pulse of antibiotics in the first few days of life. So here we're just looking at the principal coordinates analysis at day 52. This is 42 days after the single pulse of antibiotics. Here are the control mice. Here are the mice that got 1 pulse. Here are the mice that got 3 pulse. There's no normalization of the microbiota after that single pulse of antibiotics up to this period of time. These mice now are sexually mature. We asked, what's the effect of the antibiotic on gene expression in the host? So here we're looking at a heat map of work looking at gene expression in the ileum. This is from the nanostring encounter assay. In this unsupervised hierarchical clustering, in black are the control mice, and in color are the mice that received 1 or 3 courses of antibiotics. Basically, what we see is the big distinction is between the controls and the antibiotic group. Again, we found out of 500 immune and inflammatory genes in the intestine, we found that 40 days after the antibiotic exposure, there were still 63 genes that were significantly perturbed with the 1 antibiotic course. When we look at specific genes like the polymeric immunoglobulin receptor gene, we see an effect with 3 pulses or even 1 pulse. When we look at secretory IgA in the intestine of the mouse, we see after 1 pulse, it's down. After 3 pulses, it's down more, but even with 1 pulse, it's down at day 40. They're not recovering. Here's Th17 cells. They're down substantially after a single pulse of antibiotics. So we see big effects on the host from the antibiotic. So the question is, well, before we come to the next question, let me just mention that in this experiment we had a nice control because we give the antibiotics in the drinking water that the mom drinks, and then the antibiotic is in the milk, her milk, that the pup drinks. So both of them are exposed for the same period of time, and now we're going to sacrifice the mice about 40 days later and ask what happens to gene expression in the mice. And here are volcano plots looking at ileal gene expression in the dams. So this is 40 days after they got the antibiotics. We see that there are 106 genes that are differential. But in the pups, who were exposed at the same time, there were 1,300 genes that differential. So the same kind of exposure had a much bigger effect on gene expression of the pups who do not have a mature immune system. The perturbation is greater. So the question is that an antibiotic effect or is it a microbiota effect? So to address that question, Victoria gave the antibiotics at the same time, day 5 to 10, to germ-free mice or not, and in parallel gave the antibiotic to conventional mice or not, and again sacrificed the mice 40 days later. So here we're looking again at the nanostring data, unsupervised clustering. These are the normal mice, conventional mice, no antibiotics. They're clustering all by themselves. We can also see it here on this PCOA. Here are the germ-free mice. They form a single cluster whether the mice are receiving the antibiotic or not. And the conventional mice that receive the antibiotic, they're clustering with germ-free. And we can see it here. Here are the germ-free mice, antibiotic or not. Here's conventional mice with antibiotics. Here's conventional mice without. We see the conventional mice have an effect on splenocytes and also Th17. They have an effect, but the germ-free does not. So this series of experiments provides evidence that it is the altered microbiota, not the antibiotic, that is producing the effect. Finally, in this series of experiments, we asked, is the antibiotic-altered microbiota sufficient to affect immunity? So Isaac Engstrand, who was visiting us from Karolinska, did an experiment in which mice received antibiotics or not. They were sacrificed 2 days later, and now their microbiota were given to recipient mice that were followed for the next 77 days. They were germ-free. They were conventionalized. Here we're looking at immune phenotypes, T-cell populations in the spleen, down 77 days later, fecal IgA. Here's the mice that received the normal microbiota. They have the normal development. Those that receive the perturbed microbiota, their development is slow and doesn't reach the same level. So we have evidence that some of the immune phenotypes that the microbiota is sufficient for the transmission of the immune phenotypes as well. So these are all markers. So we also wanted to look at it a little further in a disease model. So Seren Erdogdu from Ankara in Turkey did an experiment where the mice received antibiotic or not from day 5 to 10 of life, and then 15 days later they were given an irritant of the intestine, DSS, or not. And now we asked, was there an additive effect from the prior antibiotic dosage? So here we're looking just at the weight of the mice, and the important curves are here. Here are the mice that received DSS, the control mice, after no antibiotic or after antibiotic. You can see that they're losing much more weight. Here we're looking at blood in the stool, more if they had the prior antibiotic. Here's histology score, worse if they had the antibiotic. So the antibiotic is again, enhancing the effect of the DSS. Here we're looking at a proliferation score. We see it as well. So again, we ask, is this due to the antibiotic sticking around, or is it due to the altered microbiota? So we gave mice antibiotics or not. 40 days later, we sacrificed the mice or not. We sacrificed the mice that received antibiotics or not. We gave them to germ-free recipients and then gave those mice DSS. Here we're looking at the recipient mice that got DSS after the control. There's their weight curve. They lost more weight if they had gotten antibiotics 40 days earlier. They had more blood. They had higher histology scores. So we have evidence that the antibiotic is affecting their susceptibility to an inflammatory process 40 days later. The final model, which I don't have time to tell you about, was work that Alexandra Livanos did looking at whether antibiotics promote type 1 diabetes. Her hypothesis is that they would. And she tested this hypothesis in NOD mice that spontaneously developed type 1 diabetes. She hypothesized that the antibiotics would change the gut microbiome, change intestinal gene expression, change T-cell subtypes, and lead to enhanced type 1 diabetes. She got her PhD and she showed all all aspects of this hypothesis. So let me just conclude with some general slides. Our hypothesis is that there's been a cumulative loss of microbial diversity due to changes in transmission. We've gone from a replete microbiota to a less than full microbiota early in life. That's the critical time when there are relationships built between innate and adaptive immunity. We're hypothesizing that there has been a difference in the relationship between these two, and this is changing autoimmune and inflammatory diseases and perhaps metabolic diseases as well. So another way to look at it is that we have been, with each generation, we in the US and perhaps in Germany, we're stepping down in our diversity. But there are countries that have developed more recently, like China, for example, or India, but their steps are bigger. They're going down, they started later, but they're going down perhaps faster, and other groups that are just beginning the decline. This is where we were in 2016, and the question is, where are we going to go from there? Are we going to have continued decline of the microbiota? Are we going to arrest the decline? Or are we going to have reversal through restorative steps? So I'm predicting that the medicine of the future is that pediatricians are going to examine babies and examine babies' diapers. And they're going to ask, which microbes do these babies have? Which global microbes that all babies should have, or which personal microbes should this baby have based on their genotype and other markers? And if the baby doesn't have them, then the doctors of the future are going to give those babies those microbes, perhaps to drink, and ultimately assess whether this is working to optimize their later risk of disease. And perhaps we will also have to drug the microbiome, not just give particular agents. Those could be with prebiotics, for example. So in concluding, I just want to recognize the many people who've done the work I've talked about today. I've highlighted the work of Il-Sung Cho, Laurie Cox, Yael Noble, Victoria Ruiz. Zach Kurtz did a lot of informatics. Angel Schulfer on the IBD, DSS model, Surin Ergogdu. Many others helped in a number of regards. I want to finish by reminding you about missing microbes. Thomas has said very nice things about it. It's now available in German, but watch out because it's being crowded out by another book. So thank you very much for your attention.